Does chronic sleep disturbance lead to neurodegenerative disease pathogenesis? We would like to provide a simple, stable, and long-term protocol to model frequent interruptions during human sleep to address this question.
This mice minimizes handling labors and artificial bias. It can standardize the interventions among mice within the same experimental group and between different experiments to produce repeatable behavioral and pathological outcomes.
This model can be applied to a variety of disease models, such as depression and Alzheimer's disease, to study the impacts of chronic sleep disturbance in the presence of an underlying disease.
Begin by randomly assigning 3 to 5 20 to 28 gram 8 to 10-week-old, adult, wild-type male mice to CSF modeling and control cages to avoid social isolation stress. House both groups of cages in the same 21 to 23 degrees Celsius and 35 to 60% humidity modeling room with a 12-hour light-dark cycle to maintain an identical surrounding environment. Then, provide the mice with sufficient food and water, using long nozzles with ball valve tips on the water bottles to prevent water leakage upon the platform movements, and fastening the water bottles on top of each cage with a spring to avoid dislocation of the bottles during rotation.